All right, guys, so I want to do a little bit of a study with you guys. I go to college, so I want to break it down. I'm just going to read it. I'm probably going pretty fast. So, this is research methods in neuroscience. Methods on how to research the brain. So, scientists, researchers, they use structural imaging techniques, they use functional imaging techniques, they use non imaging techniques, animal techniques, and neuropsychological techniques. First ones we're going to go over are the structural imaging techniques. Think about it, a structure. When you look at something, you see the structure of it, what it looks like, and in a way, how it's built, how it looks. And so this is structural imaging techniques. Try to remember that, structural imaging techniques. And these techniques are good for finding tumors, sort of maybe autism, size of the brain, and structure of the brain. Some of the techniques used for getting images of the brain, the structural images, are X-rays, which are two-dimensional images on film. They're best for hard tissues, such as bones. Another structural imaging technique is a CAT scan or a CT scan, a computerized tomography. There are a series of X-ray style images taken by a specialized computer, hence the CT scan, the computerized tomography scan. They're better for soft tissues than X-rays are. Then you have magnetic resonance imaging, which is an MRI. A lot of people have heard of MRIs before. Their uses are powerful magnets to get an image of the brain. They're powerful magnets. And with MRIs, there's no radiation when put under these machines. Unlike the first two, with X-rays, which are 2D images on film, and with CT scans, there is some radiation involved, but not with MRIs, no radiation. Now we go to functional imaging techniques. Think about it. You go from structure to how it looks, and you go to functioning, how it works. So we're working with the brain. You have structural imaging techniques of the brain. You have functional imaging techniques of the brain. You got direct techniques and indirect techniques. So functional imaging techniques, direct techniques, there's two of them. Electroencephalography, also known as EEG, magnetic encephalography, MEG, and then there's indirect techniques, positron emission tomography, PET, and then you have functional magnetic resonance imaging, so functional MRIs. The first one was just MRIs, and now we have the same machine, but it can detect, it can notice, it can capture the functioning of the brain. And so just back up to the first one, the direct imaging techniques, the electroencephalographies and the magnetic encephalography. So encephala means the head. Encephala, that means the head, the head. Okay, so what makes what makes a technique a direct technique versus an indirect technique? So a direct technique is going to detect the the electric activity of the brain and the magnetic activity, or the magnets are used to detect the electric activity of the brain. We're going to come across it again. I'll make it more understandable. So the electrical activity of the brain, as far as indirect techniques, they detect the activity of the blood flow, which is and other things. So they detect other things and assume that these other things are affected by the electrical activity of the brain, or that they have an effect on the electrical activity of the brain. And I'll get to that a little bit later. It's called something else. It's called neurovascular coupling. How blood flow and electrical activity, like how do they relate? It's called neurovascular coupling. Release of chemicals like glutamate from neurons, which make the blood vessels expand and increase blood flow in areas with increased neuronal firing. My teacher didn't teach me this, or my professor. I just sort of asked, like, how does that work? How does a blood flow show that electrical activity of the brain? So anyway, that's the difference between direct and indirect. Direct, we directly get a reading of the electrical activity of the brain, and they also use magnets to detect the electrical activity of the brain. But indirect, like the positron emission tomography technique and functional magnetic resonance imaging technique, they're detecting the activity of the blood flow and the blood vessels. So anyway, so now we're going to go in depth with these direct techniques, the direct functional imaging techniques. So an EEG records electrical activity from electrodes that are on the scalp. So it's a machine they put on your brain, an EEG machine. So it records electrical activity from the electrodes that are on the scalp. But the thing is with electrical, this is important too. The thing with electrical activity, the electrical activity, as it's going through the skull, it smears. So it's as if like let's say a gun, when a gun or like a shotgun, or when it goes through the body, it slows down. That's exactly what happens to the electrical activity. So it smears through the skull. So there's two important things when you're trying to when you're researching, or when you're getting data of the brain and its electrical activity of your specific individual, your subject you're working with, the brain you're working with, is spatial resolution and temporal resolution. Remember, these are images. So spatial resolution and temporal resolution. Spatial, think about space. Think about think about location. Think about where. So where. Spatial is where. Temporal resolution is when. Temporal, temporary, temperature, like when. So you got where and when. That's what we're trying to get with these different techniques. Some of these techniques are a lot older when it comes to science and neuroscience. So we've gotten better at this thing. So the EEG, the electroencephalography, it records electrical activity from the electrodes that are on the scalp, but the electrical activity smears through the skull. So the spatial resolution, where is this activity occurring in the brain? How accurately can researchers figure out where the activity occurred? So the EEG, since the electrical activity actually smears when they're going through the skull, has poor spatial resolution. It's not that accurate. But temporal resolution, when the activity occurred, it has a good temporal resolution. It can tell you when it occurred, the electrical activity. But since it smears, it's hard to tell the exact location where it happened. But when it happened, temporal resolution is pretty good. How an EEG works. Again, anyway, so it says, anyway, so the good things about an EEG is that, like I said, it has a good temporal resolution. It can tell you when. It's pretty accurate when it can tell you when it occurred, the activity in the brain. Remember, this is a machine. This is a imaging technique. So it's like the pros and cons of an EEG. So it can tell you when the activity occurred, but it's not that good telling you where it occurred, right? Because of the smearing. And only the activity closest to the scalp is easiest to measure, because it's closer, and it doesn't have much of a smear. So there's some restrictions there. So with an MEG, so let's go to MEG now. Remember the magnetic encephalography. The first one was an electrical encephalography. This one's a magnetic encephalography. So every electrical field has a perpendicular magnetic field. A perpendicular magnetic field. So that means at some point they either cross, right? A magnetic. So every. Okay, so here we go. So every electrical field has a perpendicular magnetic field. So perpendicular means a 90-degree angle. It could be a cross, so we got four 90-degree angles here. It could be a 90-degree angle. That's perpendicular here. Perpendicular has a 90-degree angle. It could be a triangle. You got a perpendicular here, right here. My finger go up, the two fingers meet. It's perpendicular here. So MEG detects the magnetic field, and EEG would detect the electrical field. So an MEG detects the magnetic field. Magnetic fields don't smear through the skull. So what that tells you, it has a better spatial resolution. It's more accurate when it can tell you where the activity occurred, as opposed to the electrical field. That electrical activity smears through the skull. Magnetic fields don't smear through the skull. Pros and cons. So the pros, a magnetic and just say MEG. An MEG compared to an EEG has good temporal, can tell you when it occurred, or the EEG had a good when the electrical activity occurred in the brain. I guess we'll call it action potential. Action potential means electrical signaling. So the action potential for an MEG is good because we know exactly when it occurred, just like the EEG. And it's also good at spatial because the magnetic field doesn't smear through the skull, so we can tell where it occurred. The spatial resolution of the action potential, which is the electrical signaling, is good. The cons about it, spatial resolution is still not great because what happens is it's like a flat surface, right? Since since the magnetic field and the electrical field cross, there's still a little bit of a obstruction of the action potential. I think I could be wrong. I could be saying the wrong thing right here. I actually want to ask my teacher to break it down again. Why the MEG would it says so this MEG only measures information in the grooves or the sulci, which are the grooves of the brain. You know, the brain looks like it kind of just like doesn't really touch. There's like little gaps in between the brain. So this MEG only measures information in the grooves of the brain. It's also very expensive and not really found in a lot of places. Very expensive. It's like millions of dollars. All right, guys. So direct versus indirect imaging. So we went over the direct and indirect um, imaging techniques, right? We went over these already. So we're gonna the direct versus indirect imaging. So direct imaging techniques directly measure electrical or magnetic activity from the brain. Remember, the EEGs and the MEGs are direct imaging techniques. Indirect imaging techniques measure something else and assume it relates to electrical activity from the brain. Most of the time, that something else is blood flow. So indirect imaging techniques measure something else besides the electrical and the magnetic activity of the brain, but they measure something else and assume it relates to the electrical and the magnetic activity from the brain, which are indirect techniques. They measure blood flow. Okay, so actually, we haven't went over the indirect techniques, not entirely. So the indirect techniques, one of them is the PET, the positron emission tomography, the PET scan. Positron emission tomography. They use a tracer, they inject it into the bloodstream. Positrons are emitted from the tracer and detected by the system. It creates a map of where the positron came from as a measure of activity. Maps usually represent activity that occurred over a span of multiple minutes. Remember, we need to know when it happened as soon as possible. This usually maps out and represents activity that occurred over a span of multiple minutes. So what that tells us is that the temporal resolution is not good. Like precisely when the activity occurred, since it happens over minutes, the positron emission tomography is not that great at giving us the information of exactly when it occurred. But it is good at telling us where it occurred. It's good at telling us where it occurred. Again, the cons are it's kind of poor at telling us when it occurred because it maps the activity within. It gets like a map of it, but of minutes. What is this right here? So it creates a map of where the positron came from as a measure of activity. Maps usually represent activity that occurred over a span of multiple minutes. So it's not really good at telling us exactly when the activity occurred, but it is good at telling us where it occurred. And also there's small levels of radiation in PET scans. And here we go. Another indirect technique is a functional magnetic resonance imaging. So as you're let's say you need to get an fMRI, you need to get a functional MRI. At the same time, you can also get a regular MRI, a structural MRI of the brain at the same time. It's able to take a photo of the brain. So an fMRI is able to detect changes in the blood flow by looking at the oxygen levels of the blood vessels. So fMRI, they detect changes in the blood flow by looking at oxygen levels. You can also take a structural MRI scan while the subject is in the scanner. It allows for a nice overlay of the functional scan. So pros of this, spatial resolution is good. We know exactly where the activity occurred, the location. And it says also the temporal resolution is better than a PET scan. Um, when it occurred, the activity is better than a PET scan, but not as good as the direct imaging of the EEG and the MEG. But there's no radiation involved, unlike the PET scan. Some of the cons is scanner is super loud, only so many things you can do inside of a magnet, so there's some limitations. Other concerns for these indirect techniques, remember that they're indirect. So we're assuming that blood flow equals brain activity, but if that's incorrect, the information and the findings of the PET and fMRI, the data we collected from using these techniques would be incorrect because we're assuming that the blood flow equals brain activity. Remember, people's bodies are different, right? So how do PET scans and fMRIs work? They're indirect techniques. So they perform a subtraction analysis. They record brain activity with subjects doing what you're interested in. Compare that activity with an appropriate baseline. Many times researchers subtract out the baseline activity to be left with areas of increased, decreased activation. I guess like a normal, what your brain activity, what is it just like normal? And they take that out and just see all like the changes of what's your normal baseline activity. Yeah, I can imagine that's what that means. So remember, so how does blood flow relate to electrical brain activity? It's neurovascular coupling, release of chemicals like glutamate from neurons, which make blood vessels expand and increase blood flow in areas with increased neuronal firing. Yeah. Okay, now we're at non-imaging techniques. Non-imaging techniques, and we'll probably go to animal techniques, and then we'll just maybe do another video after, like later. So non-imaging techniques, transcranial magnetic stimulation, also known as TMS. It uses a magnetic wand to stimulate and also inhibit the brain. Magnetic waves travel through the skull easily. They don't smear. They excite the brain or they shut off a part of the brain. So it's obviously good for spatial, like where these things have occurred. It's sort of a new application, TMS, transcranial magnetic stimulation, used for many disorders, including schizophrenia, depression, Alzheimer's disease, autism spectrum disorder. So RTMS, which is probably, I don't know what the R stands for, can reduce brain activity and possibly stimulate cortical plasticity. The cortical means relating to the outer layer of the cerebrum. Uh, so again, there's something called neurofeedback therapy. Neurofeedback therapy. I heard it's really good. I read a really good book about it. How this is a scientist too. The scientist who wrote a book on trauma, how the body keeps a score. It's a really good book by Bessel Bessel Vanderkolk. Really good writer and really one of the best books I ever read. The body keeps a score. It's very popular. A lot of you guys have probably heard of it. He mentions a technique called neurofeedback therapy, and he said it does amazing things. They're able. I think this transcranial magnetic stimulation is able to redirect the brain activity, parts of the brain that fire. They're able to kind of just get rid of it and maybe push it to redirect that electrical brain activity to somewhere that it's better suited for an individual with these schizophrenia or depression, traumatic situations, PTSD, stuff like that. All right, so animal techniques, they're single cell recordings. Animal techniques, single cell recordings. Obviously, they're an invasive technique. You have to drill a hole in an animal's brain. It records activity using a tiny device known as a microelectrode. Allows researchers to determine the specific function of a neuron. The pros and cons of single cell recordings: excellent spatial resolution, which they could tell you exactly where the electrical activity occurred, and also they could tell you when it occurred. The temporal resolution is really good as well. But the cons, it usually misses what is happening everywhere else in the brain. Remember, it just focuses on one neuron and is damaging to the brain of the subject, is damaging to these animals that we use. That we So single cell recordings, they're animal techniques. And sometimes recordings are likely extracellular and gathers activity from a few cells. It's not precisely only one cell being recorded. And now we're kind of going to go on to neuropsychological techniques, but you might say that for another video because we're already on 23 minutes. And yeah, that's pretty good so far. I actually have class tomorrow, so whatever I'm going to go through over tomorrow will probably be included to what information that I actually have left. Yeah. So we're going to get into neuropsychological techniques, which these are pretty interesting because you got surgeries and little different surgeries they used before, different procedures, different drugs they use to actually shut off certain parts of the brain. Also, like the ethics of human subjects and non-human animal subjects. Then we're going to get to actually how the development of the nervous system, how we come from the embryo and how our body actually forms and how our neurons are given to us. And so what am I going to learn? Oh, and then we're working on things like the central nervous system and how it's made, like the spinal cord, the brain, the brain stem, the forebrain, the midbrain. We got the cerebrum, which is the cerebral cortex, the spinal cord and what it's made of. Yeah, or there'll be another video for tomorrow. Anyway, y'all, take care.